Today we're going to be talking about reproduction in fungi, specifically the bread mold Rhizopus, which belongs to the phylum Zygomycota. Now, Rhizopus can reproduce both asexually and sexually. It wants to reproduce asexually when conditions are good, meaning there's plenty of water, plenty of food, plenty of nice temperature. In that case, when conditions are good, why not make genetic copies of yourself so that those individuals will do just as well as you're doing in that environment? However, when conditions change and are not so great, not enough water, not enough food, maybe the temperature is not to our liking, in that case, we want to make genetically different individuals in hopes that some of those individuals will be better adapted to that environment than we are. So let's go ahead and get started. We're going to get started with Mycelium. Now, Mycelium is simply a whole slew of hypha, and hypha is an individual fungus. That's an individual fungus cell. So Mycelium, we've got a whole slew of individual fungi cells, and the mycelium is going to give rise to a spore-producing structure. This spore-producing structure is then going to produce asexual spores. And they're going to be N. Now again, I say asexual spores because these spores, these reproductive cells, are going to be genetically identical to the parent that they came from. These asexual spores, if they fall on a suitable habitat, suitable substrate, are then going to go ahead and they're going to grow into a mycelium. That occurs when conditions are good. That is asexual reproduction. Good conditions. But what if conditions aren't so good? Then we want to think about reproducing sexually. So again, we're going to start with our mycelium. And the mycelium is going to produce a structure that looks like this. Kind of like a big H. Now these are two individual hypha here. So here's one hypha. And here's another hypha of another individual. They look the same on the outside, but genetically they're going to be different from each other. So let's call this plus, the plus mating type. Let's call this one the minus mating type. And all this means is that these two hypha, these two fungi, are genetically different from each other. I could have called this one the minus one, and I could have called this one the plus one. Doesn't matter. All it means is that they're genetically identical from each other, are genetically different from each other. Now let's go ahead and denote the nucleus of this hypha with red, and the nucleus of this hypha with blue. And both of them are going to be N. This is called the heterokaryotic stage. In this stage, the cells are actually fused together. Cells fused together through a connection known as a conjugation tube. These two hyphae can actually sense each other through the use of chemical attractants called pheromones. These hyphae then produce outgrowths which then connect to each other and form a conjugation tube. It's through this tube that the two nuclei are going to come in contact with each other. Now the next stage is called karyogamy. With this stage, again, we're going to draw this H-shaped figure. But this time we're going to show the two nuclei fusing together. So the nuclear envelopes of these two nuclei have fused together, and the chromosomes are now in contact with each other. So in a sense, this is a single nucleus. So we'll denote it as 2N. Now what's going to happen here is, in order for meiosis, and that's what sexual reproduction is all about, meiosis, in order for meiosis to take place, you have to have two copies of each chromosome. And that way crossing over or exchange between pieces of DNA can occur. So with karyogamy, what we're going to have there is the nuclei fuse. That's karyogamy. And karyo means nucleus. So the nuclei actually fuse together. Here we have the heterokaryotic stage, and again, hetero means different, and karyotic means nucleus. So the nuclei are still different from each other. They haven't come in contact with each other yet. From this stage, we're going to go ahead and we're going to produce a spore-forming structure. And this spore-forming structure grows out of this conjugation tube, and it produces sexual spores. And they have the N number of chromosomes, meaning they have one copy of each type of chromosome. And I like to call them sexual spores because these spores are genetically different from each other, and they're also genetically different than the two parents that they came from. That's very important. These sexual spores will then continue to grow into a mycelium. Now the adult fungus, the mycelium in this case, is going to be genetically different than the parent it came from. And again, this is important. If conditions are bad, we want to produce kids that are genetically different from the parent, in hopes that some of those kids will be better adapted for that new environment than the parent was. So this is going to be sexual reproduction. And it occurs during poor environmental conditions. And that's it for reproduction in fungi.